Respected chairpersons and my dear colleagues, very good afternoon to all of you and greetings from the debate team. I am thankful to Dr. Basant and his team to give me an opportunity to debate on this topic on this platform. We are discussing if genetic testing should be done for all pediatric cancer patients, and I shall be speaking for the topic. So we all understand that genetic testing is a whole new world of insights. These two words, genetic and testing, seem very small, but when you go into the intricacies, it is a mega subject. So here I am trying to depict the vastness of the role of genetic testing. When I was preparing this topic, I was reminded of my MBBS days. I did my MBBS from King George's Medical University itself. So that was the time when my parents used to give me some 2,000 rupees per month for all my expenses. And Harrison used to cost around 16,000. So we went to a local market behind Hazratganj and I could find a local copy of the book. Now, the challenge was, once I got the book, if it has the same content as the original Harrison. So here we have the gold standard, which is the reference human genome, and we have the patient who has the disease, and we are trying to identify his genetic composition. And through this analogy, I will just go through the basics of genetic testing to help you understand what all is possible. Because unless we know what all is possible, we don't know why it is important. So while I was comparing these two books, I realized that this local copy had two set of all the pages. So technically it was double the Harrison and everything was in duplicate. So this is aneuploidy, basically triploidy, tetraploidy, and you can detect with the help of chromosomal analysis or karyotyping. The second scenario, the entire book was fine, but there was a chapter which was there in duplicate in the local copy. So this is analogous to extra copies of the chromosome, and this again is possible with karyotyping. Now I realized, no, it's not a problem with chapter also. It is a paragraph in the local copy, which is different from the actual book. So this is analogous to large deletions and duplications, and you can identify them with chromosomal microarray analysis. If just a line is different, that is analogous to small deletion or duplication, and you can find out with a copy number variant analysis. So these are the different types of genetic tests available. Now, there's not a problem with the line also. There's a single word, say Goel. It's instead of G-O-E-L, it is in G-O-O-L. So there is a single alphabet has been replaced, and this is a single nucleotide polymorphism, and you can identify with the help of next generation sequencing, which is whole exome sequencing or whole genome sequencing. Now, I have to appear for a terminal examination, and I realized I don't need to compare the whole book. I am concerned with a particular chapter which is included in this syllabus. So this is a disease panel which can be identified with the help of targeted sequencing. Now, my roommate, he was like, yeah, this book is comparable, but somehow I don't like the page quality, the typesetting, or the binding of the local copy. So this can be compared to DNA methylation or histone modification. Technically, that is the epigenetic study. And finally, after I read the local books, my teachers were concerned that this guy did not read original Harrison. Now, will this local book bring about the same change in his personality, his knowledge database, his attitude, etc.? So this is about gene expression or transcriptomics. So with this background, so what are the indications of genetic testing? So it's a very wide domain. Like I said in the beginning, we need genetics for clinical indications. We need genetic testing for research purposes. We need it for public health. We need it for ethical, legal, and forensic concerns. And finally, cross-population studies. So cross-population can be dealt with separately or as public health. We will focus ourselves largely on the indications related to pediatric cancer. The first one is clinical, which is the widest domain. So my colleague, Dr. Manish Pathak, would be talking in detail about this. But just to give an overview, you can use genetic testing in pediatric cancers to make a diagnosis of the disease. You can use it to identify the subtype of the disease. You can use it for risk stratification. You can use it to identify which would be the best or better therapy for the disease. You can have therapeutic targets identified, and so on and so forth. So I leave this part to Dr. Manish Pathak. Then you can use it for research purposes like genetic discovery, clinical trials, tumor heterogeneity. We'll talk about it subsequently. Public health as in population screening programs. So to cut short, I just summarized all these into these points. That what are these are 7 indications I mentioned, and just a brief about all these points. So basically what is happening is India is a vast country. It is geographically vast. We have peoples of different ancestries. So it is ethnically vast. The climate, the environment is different. So overall, you cannot club India into a single term when you're talking about this. So one thing is one size fits all. This does not apply to India. Our research which has come from the Western world will not be applicable to us in an as-is manner. Secondly, we have realized, we all will agree with that, in most of the Western studies, our genetics is largely underrepresented or not represented at all. And finally, being such a huge economy, being an economy with such a huge population which stands out in the world, we should have a genetic database of our own markers. So our genetic database and the consequent genetic testing, it must reflect the unique needs and diversity of Indian population. Somebody may claim that my ancestry is common, but over the generations, things change. That's what we'll discuss. So this diagram is trying to represent India as a whole, but then you see that there are people from different parts of the country with different ethnicities, and they have different databases. So what has happened is that genetic markers from the Western world, they are not able to encompass comprehensively the diversity that we encounter in our population. And for those people, maybe somebody is coming has a common ancestry, had settled down 5 generations ahead. He has undergone what we technically call as genetic distancing. So his genetics may not work the same way what it used to work 5 generations earlier. So yet another creation wherein I am trying to depict the role of environment and epigenetics. So basically diversity within unity. So what happens is the same genetic markers in the genome of 2 individuals, they may behave differently when their effect or the outcome is modified because of the local environment or the epigenetic influences. Besides this environment or geography or epigenetics, we have also realized that the role of socio-economic factors also is again very, very important. So I'm sure none of us would disagree that India needs a genetic database which would become culturally relevant and which is culturally relevant to our society and which would lead to capacity building. And that is the way. So other than clinical part, I have emphasized on the other aspects why we actually need genetics. We need genetic testing in our patients of cancer. I have illustrated the role of genetics briefly with the help of one tumor, that is hepatoblastoma. We could have chosen Wilms tumor. I think Dr. Manish is talking about Wilms tumor. I chose hepatoblastoma because this particular tumor encompasses different genetic tests into one. So we all identify these 2 syndromes, and we know that hepatoblastoma is one of the manifestations in both of these syndromes. We know that the genetic basis of the etiopathogenesis of hepatoblastoma, it lies in the WNT beta-catenin pathway and partly into the canonical beta-catenin pathway. So we have seen the role of DNA methylation in hepatoblastoma, that is epigenetics. We know the role of tumor suppressor gene promoters like APC. We know the role of hypomethylation of so-called oncogenes leading to their activation. The other aspect of epigenetics is histone modification like H3K9 deacetylation and inhibition of SDAC. Both of these have again been implicated. So this was about epigenetics. We know the role of non-coding RNAs in hepatoblastoma. And these are the RNAs which have been described. So this was the clinical part, the research part, the community medicine or the population studies part. And I think a combination, a balance between all these aptly says that we do need to perform genetic testing in our patients with cancer.